All right, welcome back to the deep dive. Today we're diving into an area that's pretty hot right now in research, and what's about aging. And this particular study that we're going to be talking about today really caught our eye, and I know a lot of you listeners out there flagged it for us, so we definitely wanted to take a look at it.
Yeah, this one's a really interesting one. So, we're always on the lookout for studies that kind of uncover these hidden sort of connections in biology, and this one is about a protein called AP2A1, and the question that we're going to be asking today is how this, kind of seemingly, not a very well-known protein is actually linked to how our cells age, and the process is called cellular senescence.
Right. So just to give everybody a background on why this is so important, cellular senescence is basically like the wear and tear that happens on our cells over time. And it's really at the heart of all of these age-related issues that we face. And so the more we understand about it, the more we might be able to come up with interventions or ways to address those issues. So we're going to be doing a deep dive today on this study and really trying to understand the proposed role of AP2A1 in this whole process. It's like we found a new suspect in this whole aging mystery, and we're gonna comb through the evidence and see if we can connect the dots.
All right, so to lay the groundwork a bit, we're talking about cellular senescence, and think about it this way: as our cells divide and do their jobs throughout our lives, they undergo changes. They don't stay as youthful and efficient as they used to be, and they start to kind of show these signs of wear and tear, and that's what we call senescence.
And that's not just a cosmetic thing that's happening at the cell level, right? It has real implications for our overall health.
Precisely. It's not just about wrinkles on the outside. This cellular aging contributes to, our tissues and organs starting to decline as we get older, and it's a contributing factor in a whole range of age-related diseases. So getting a handle on what's going on inside these individual cells is really, really crucial. Now, the study also delves into these fascinating structures within cells called stress fibers.
Stress fibers, okay. What are they and why should we care about them?
So imagine a cell has this internal framework, a bit like scaffolding you'd see in a building, and stress fibers are a key part of this framework. They're mainly made up of a protein called actin, and they help the cell keep its shape. They allow it to move around and interact with its environment. They're basically fundamental to how the cell mechanically operates and responds to forces.
So they're not like static, rigid structures. They actually have some dynamism to them, yeah. They're changing as the cell's doing its thing.
Exactly. In young, healthy cells, these stress fibers are really flexible, very adaptable. But as cells age and become senescent, these fibers tend to get thicker and they lose some of their flexibility. It's like the scaffolding in our analogy starts to become a bit more rigid. And this change in the physical structure of the cell isn't just something we can see under a microscope. It actually impacts how the cell functions and how it communicates with its surroundings.
Okay, so we're starting to build a picture here. Cells age. Part of that process involves these stress fibers becoming less flexible, and now this is where our intriguing protein AP2A1 comes in. What do we need to know about this protein?
Well, what's interesting is AP2A1 hasn't really been on the radar in the aging field until recently. It's mostly known for its role in endocytosis,
Hmm.
and that's basically the process by which cells engulf substances from their surroundings, taking in nutrients or signaling molecules, things like that. And AP2A1 is essential in forming these little bubble-like structures called vesicles that bring these substances into the cell.
So it's kind of a surprise to find it potentially linked to something as complex as aging, right?
Yeah, exactly. It's been implicated in some other conditions, but its connection to the overall process of cellular aging wasn't really clear until this study came along. And that's what makes this research so intriguing.
So tell me what they found. How did they connect AP2A1 to these aging cells and their stiffer stress fibers?
Well, they used human fibroblast cells. That's a type of cell that's commonly used in lab research, especially when studying connective tissues. And they found something pretty remarkable. The levels of this AP2A1 protein increased significantly in these cells as they became senescent. And it wasn't a small increase either. It was a pretty substantial change.
Okay, so older cells have more of this AP2A1 protein, but where exactly was this extra protein located within the cell?
Now, here's where it gets really fascinating. In the young, non-senescent cells, AP2A1 was pretty evenly distributed throughout the cell. But in the older cells, they observed AP2A1 specifically accumulating along these stress fibers, almost as if it was deliberately attaching itself to these structures.
So we've got these thicker, less flexible stress fibers in the older cells, and now we're seeing this protein AP2A1 sort of clinging to them. That sounds like more than just a coincidence, right?
That's exactly what the researchers are proposing. The idea is that this interaction between AP2A1 and the stress fibers might actually be contributing to or even reinforcing the changes that happen in these fibers in aged cells. Like AP2A1 is almost becoming part of the machinery that's keeping that scaffolding rigid. And what's really compelling is that they saw this same pattern no matter what triggered the cells to become senescent in the first place, whether it was just from the cells dividing over and over again or exposure to certain chemicals, even radiation, the results were consistent. And that consistency really strengthens the argument for a connection here.
Okay, so we're seeing this correlation more AP2A1, especially on stress fibers in these older cells. But correlation doesn't always mean causation, right? How did they determine if AP2A1 was actually driving these changes?
Right. That's where the researchers took things a step further
Mm.
and did some really elegant experiments to explore that causal relationship. They did what I like to call the two-way street experiments, and they're really convincing.
Okay, two-way street. What do you mean?
Okay, so first they tried to kind of rewind the clock on the older cells. They used a technique called silencing RNA. So basically, silencing RNA is a way to specifically target the gene that produces AP2A1 and turn down its activity. So it's like they're lowering the volume on that gene, and that allows them to see what happens when there's less of the AP2A1 protein being made. And they found some really striking results.
So what happened? Did the cells suddenly start acting young again?
It wasn't a complete reversal, but they did see some noticeable changes. Those thicker stress fibers became less prominent. They started to look more like the ones you'd see in younger cells. And maybe even more importantly, the levels of those classic markers of senescence, things like P53, P21, and SA-beta-galactosidase, which are like telltale signs that a cell has entered that age state, they all decreased.
Wow, so reducing AP2A1 in these old cells seemed to rejuvenate them in several key ways. That's pretty convincing. It's not just that old cells have more AP2A1, it seems like that extra AP2A1 is actually helping them stay old. And on the flip side, too much AP2A1 in young cells is nudging them towards that aged state. That's a big deal, right?
It really is. And they didn't stop there. They did the opposite experiment too. They took young cells and artificially increased the amount of AP2A1 they were producing.
And did those young cells start to show signs of aging prematurely?
They did. The young cells started to exhibit some of the hallmarks of senescence. They got larger, they developed more prominent stress fibers, and the levels of those same aging markers, P53, P21, SA-beta-galactosidase, the ones went up. It's like they were pushing these young cells towards an aged state just by increasing the AP2A1 levels.
Wow, that two-way relationship, reducing it, seems to make old cells act younger. Increasing it makes young cells show signs of aging. That really solidifies the argument for AP2A1 playing a key role in this whole aging process, doesn't it?
Definitely. It moves beyond just an association to a more causal link. AP2A1 isn't just a passive bystander in the aging process. It seems to be actively involved in driving those changes we see in senescent cells, especially those related to the structure and function of the stress fibers.
So if these findings are true, are we talking about a potential new target for future anti-aging therapies? I mean, could we develop drugs or treatments that manipulate AP2A1 levels to slow down or even reverse cellular aging?
It's certainly an exciting avenue for future research. But like the researchers themselves acknowledge, we need to keep in mind the limitations of this particular study. It was conducted on just one type of cell, fibroblast, in a very controlled lab environment. The human body is incredibly complex, right? There's so many different types of cells, and what we see happening in a petri dish might not perfectly translate to a living organism.
That makes sense. And you said earlier that AP2A1 has this crucial role in endocytosis, which is a fundamental process for all cells. Could targeting it broadly have some unintended consequences? We don't want to mess with something that's so essential for basic cell function.
That's a really important point. AP2A1 is involved in this very basic cellular mechanism, how cells take in essential molecules. So just trying to get rid of it altogether could disrupt all kinds of vital processes. We had to be careful about thinking of it as a simple on-off switch for aging.
So maybe the focus shouldn't be on completely eliminating AP2A1, but rather on understanding its specific role in senescent cells or its downstream effects on those stress fibers.
Right.
Like finding more targeted approach.
Exactly. The goal would be to understand how AP2A1 interacts with those stress fibers in aged cells, and if there's a way to disrupt that interaction without interfering with its other essential functions. Perhaps there are specific molecules or pathways that are influenced by AP2A1's presence on stress fibers that we could target instead.
It's almost like this research has opened a brand new door, hasn't it? A whole new avenue for exploration. I mean, the idea that a protein that's primarily known for one basic function could also be so intertwined with the aging process is pretty remarkable.
It really highlights how interconnected everything is within a cell. It's a web of interactions.
Mm.
And this study really underscores that point, that the physical structure of a cell, like something as seemingly basic as how its internal scaffolding is arranged, isn't just about shape. It has profound effects on how that cell behaves and potentially how long it can function effectively. It's like biology throws you a curveball sometimes. You think you have a protein all figured out, and then turns out it has this whole other side gig going on.
So, just to bring it all together for everyone listening, what we've learned today is that AP2A1 is a newly identified protein that seems to have a significant role in cellular aging, and it appears to do this by influencing the structure of those stress fibers inside our cells.
And what's really surprising is that we previously thought of AP2A1 as mainly involved in endocytosis. Now, this study suggests it has this other important role in the aging process.
And if these findings are confirmed by more research, in different types of cells and eventually in living organisms, it could lead to some really exciting new strategies for targeting age-related decline at a cellular level. I mean, if we could find ways to tweak AP2A1 levels or how it interacts with those stress fibers, we might be talking about actually rejuvenating cells. That's kind of the long-term goal here, right? It's pretty mind-blowing to think about.
Absolutely. This research raises some fascinating questions. How many other seemingly small changes at the cellular level, things that we might dismiss as just minor structural adjustments, could actually have profound effects on the overall health and lifespan of our cells and ultimately ourselves? What other proteins currently lurking in the shadows of aging research might hold crucial clues to understanding and potentially influencing these fundamental biological processes? That's something to ponder and maybe explore further on your own.
It is. Well, that's all the time we have for today's deep dive. Thanks for listening, and we'll see you next time.